Okay, good afternoon. Can you hear me? Good. So there are 150 students in second year of dentistry, and you are now 25. 25. One of you is inactive. So 24, right? What a dedication. Okay, so let's start. Today is our second lecture about anatomy of the brain. And if you remember, at the previous lecture, we were talking about all the parts of brain except cerebral hemispheres. So today we must complete the brain finally and CNS as a whole, because I think now nearly all the groups have completed CNS and maybe even started peripheral nervous system. Today we will discuss the following issues. End brain first. End brain is telencephalon, then conductive tracts or conducting pathways, meninges and intermeningeal species of the brain, and production and outflow of cerebrospinal fluid. So this will be the main points which we will talk about today. And as always, when we discuss part of the brain, we will start with embryogenesis. So end brain, it is English name, and telencephalon, it is Latin and Greek. So at the stage of 3 brain vesicles, end brain is developed from prosencephalon or forebrain, and at the stage of 5 brain vesicles, at the secondary brain vesicle stage, it is developed from telencephalon. You remember that prosencephalon is divided into telencephalon and diencephalon. So cerebral hemispheres, they are developed from telencephalon. End brain includes the following parts. First of all, these are cerebral hemispheres, hemispheria cerebralis, then callosal commissure, corpus callosum, fornix, and anterior commissure. So cerebral hemispheres, these are the main, the largest part of the end brain, yes, and callosal commissure, anterior commissure, and fornix, they contain white matter fibers which connect hemispheres with each other. We'll later say today at the lecture that they are formed by commissural fibers. And cavities of end brain are lateral ventricles. Like you know that each part of the brain has its own cavity. We were saying that cavity of diencephalon, it is 3rd ventricle, and cavity of the rhombencephalon is 4th ventricle, and so for the midbrain it is cerebral aqueduct. For the end brain, these are lateral ventricles. They are paired, one for right hemisphere and one for the left. So here you can see a cerebral hemisphere. You have to be able to distinguish right and left cerebral hemisphere. It's actually not that much difficult. There are 3 surfaces in cerebral hemispheres: superolateral surface, that is convex; medial surface, that is flattened; and inferior surface, it is flattened in anterior side and it is concave in the posterior side here. And what else allows us to distinguish right and left cerebral hemisphere? It is a presence of some gyri and sulci. So before we talk about it, we should say that surface of cerebral hemispheres is not smooth, like in cerebellum. You remember we were talking about it. And there are some elevated and some depressed areas. So elevated areas, they are gyri, yes, and depressed areas, they are sulci. Sulci and gyri, these are plural, and sulcus and gyrus, it is singular. So sulcus is a depression or groove in cerebral surface. So here in this diagram, you can see this is sulcus, And some grooves, they are deeper than the others, and they are named fissures. So a deep groove, it is usually called a fissure. Gyrus is an elevated area on cerebral cortex. So gyrus is located between two sulci. Sulcus is located between two gyri. Yes, it's obviously. And you also have to know why this surface of cerebral hemispheres is not smooth. And it is due to the presence of cortex. Like in cerebellum, we were saying that gray matter is presented by two types of structures. That is nuclei, and nuclei, they are present in every part of CNS. And there is also cortex. Cortex is present only in cerebellum and in cerebral hemispheres. And cortex, it surrounds cerebellum and cerebral hemispheres from outside. So the larger surface area, the more cortex is. And as you understand that this cortex, it is a gray matter, the more gray matter, the more neuron bodies, the more brain is developed. That's why brain of humans has the largest amount of gyri and sulci, and sulci are the deepest in the humans. If we take, for example, brain of apes, it will not be that much uneven, let's say so. What you studied at school is that cerebral hemispheres contain 4 lobes. Yes, it is frontal lobe anteriorly, parietal lobe in the middle, and an occipital lobe posteriorly, and below there is temporal lobe. Later we will say that actually there is additional lobe or 5th lobe, that is insula. We will see it later. So sulci, the main sulci of cerebral hemispheres, which allow us to distinguish right and left cerebral hemispheres. First of all, here on the superolateral surface, we can see central sulcus, sulcus centralis, that separates frontal lobe from parietal lobe. Below there is sylvian or lateral sulcus, sulcus lateralis, that starts posterior-superiorly, and it moves anterior-inferiorly, and it separates frontal and parietal lobes superiorly from temporal lobe inferiorly. Here, from superior aspect, we can see longitudinal fissure, fissura longitudinalis, that separates right cerebral hemisphere from the left one. And below there is corpus callosum in the lower part, a lower boundary of longitudinal fissure. It joins corpus callosum. On the medial surface of the cerebral hemispheres, we can see parieto-occipital sulcus, sulcus parieto-occipitalis, that separates parietal lobe from the occipital lobe. On the superolateral surface, we cannot see this groove, just an imaginary line, like a continuation of parieto-occipital sulcus, but there is no such sulcus here. So these are the main sulci which helps us to identify, to distinguish right and left cerebral hemispheres. Yes, and I told you there is actually the 5th lobe, that is insula. If to translate insula from the Latin, it will be small island, islet, yes? Like insulin, you remember, insulin. It is a hormone that is produced by islets of Langerhans in pancreas, yes? So, yes, it's small island. So this lobe, it is located in the depth, it is surrounded by all the other lobes, and we can see it only when we separate from each other, like pull out from each other edges of lateral sulcus. So this is insula. So technically it is the 5th lobe. It is about external structure. In the internal structure, like in all the other parts of the brain, we can distinguish white matter and gray matter. And like Yes, like I have already told you, gray matter is presented by two structures. It is cortex and basal ganglia. So we'll talk about basal ganglia a little bit later. Actually, we should say that it is a misnomer. Yes, it is a wrong name. It will be more correctly to call them basal nuclei, but historically they received such a name. But in modern books, in new books, they are named basal nuclei. So cerebral cortex has much more complicated structure than cortex of cerebellum. Now I'm talking about neocortex, so the newest cortex. If you remember, at the previous lecture, we were discussing structure of cerebellar cortex, and we were saying that it has only 3 layers. Here, neocortex, the newest cortex, that occupies the largest part of the cortex of cerebral hemispheres, has from 5 to 8 layers. There are some parts, some regions, which are presented by paleocortex and archicortex, which have less amount of layers, but they occupy insignificant regions. Like we will talk about it later. For example, olfactory brain, limbic system, like cortical part of limbic system and olfactory brain, yes, it is presented by paleocortex. But most part, the largest part of cortex, it is neocortex that appeared phylogenetically recently. So you have to know the structure of cerebral cortex. Typically, 6 layers are distinguished. So the first layer, we start from outside and we are moving inward, in the inward direction. So the first layer, it is molecular layer that is presented by a few scattered neurons and many dendrites of pyramidal neurons, glial cells. Then there is external granular layer that is presented by small pyramidal neurons and numerous stellate neurons. And the third layer, it is external pyramidal layer that is presented predominantly by small and medium-sized pyramidal neurons. I want to tell you that all of these 3 layers of neocortex, from the first to the third layer, they act as intermediate neurons between 4th and 5th layer. Because 4th layer, it is internal granular layer. Here there are different types of stellate and pyramidal neurons. Cells of the 4th layer of cortex, these are the cells where all the ascending tracts end. Spinothalamic, bulbothalamic, nucleothalamic, all of these tracts, the axons of third-order neurons, which are located in the thalamus, they end here in the internal granular layer. Internal pyramidal layer, that is the 5th one, that contains large pyramidal neurons, and in the primary motor cortex or frontal lobe, the 5th layer contains bed cells. These are giant pyramidal cells. That is the layer from which all the descending tracts, pyramidal tracts, start. Actually, they are named pyramidal because they start from these large and giant pyramidal neurons. And so information via the ascending tracts comes to the 4th layer of the cortex. And then it is analyzed. And then signal about the response is formed. And this signal should be transmitted to the descending tracts. So from the 4th layer, information should somehow come to the 5th layer, and it goes through these layers from the 1st to the 3rd, which act as intermediate neurons. And so the 6th layer, it is multiform or polymorphic cell layer. It is presented by few large pyramidal neurons and many small spindle-like pyramidal and multiform neurons. So that is a structure of neocortex. I told you there are not always 6 layers. Sometimes they are 5. For example, in post-central gyrus, in precentral gyrus, there is no internal granular layer at all, so they are 5. And in postcentral gyrus, internal granular layer is divided into 3, so they are 8 in this case. So from 5 to 8 layers, but typically in most parts they are 6. And all the whole cortex does not perform the same function. So cortex, each part of cortex, each region of cortex is responsible for its own functions. It was discovered by a neurologist, neuroanatomist, whose name was Brodmann. And with Brodmann, he divided the whole cortex of cerebral hemispheres into 47 regions. And these regions are known as Brodmann areas. Neurologist, if you decide to become a neurologist or neurosurgeon, you will have to know this scheme, this division very well. For us, we have to know just the main cortical centers. So what you have to understand is that cerebral cortex is not homogeneous. Like functionally, it's not homogeneous. When there is any injury or trauma in a particular region of cortex, there are particular symptoms. Particular, yes. For example, if there is any injury in precentral gyrus, then person will suffer from paralysis. And if there is any injury in the postcentral gyrus that is located behind central sulcus, person will suffer from loss of sensation. Why is it so? We'll talk about it right now. So yes, first of all, let's talk about these two main projectional centers in the cortex, which are located around the central sulcus. So all the centers in the cortex, they are divided into projectional, which are formed during intrauterine period. So they exist from the birth of a person, and associative centers, which are formed later with the time. They do not exist in newborns. So projectional centers, they exist in newborns, but associative centers, they do not exist. A human acquires them with the time. So projectional centers are named projectional because they are connected by means of projectional fibers with lower parts of CNS. Associative centers are named associative because they are connected by means of associative fibers with projectional centers. So there are no direct connections between associative centers and lower parts of CNS, just via the projectional centers. Yes? Yes. Okay, so two of the most important projectional centers are located in precentral gyrus, that is located in front of central sulcus and belongs to the frontal lobe, and postcentral gyrus. Sorry. Алло. Да, здравствуйте. Ну, сейчас лекция у меня идет. Да, хорошо, ладно. Спасибо. Okay, so let's continue. And postcentral gyrus, that is located behind central sulcus, and it belongs to the parietal lobe. So these are centers, precentral gyrus. It is the main somatomotor area. So all the descending tracts, corticospinal and corticonuclear, about which we have already talked actually, yes, they start from here. Postcentral gyrus, it is the main somatosensory area. So information about all types of general sensation, touch, pressure, pain, temperature from all parts of our body comes here. And arrangement of the neuron bodies, which receive information from different parts of the body in postcentral gyrus, and which send impulses to different parts of our body from the precentral gyrus, it is quite specific. So fibers which come from lower limbs and start and go to the lower limbs, they occupy upper parts of post and precentral gyrus. Then those fibers which are connected with middle part of the body, like upper limbs and trunk, they occupy middle part of this gyri. And fibers which are connected with head and neck regions, they occupy lower parts of this gyri, precentral and postcentral gyrus. Such an arrangement is known as sensory and motor homunculi. Homunculus means small human, small human. And here you can see it. Not only arrangement is interesting, but also part of the cortex that sends or receives impulses from different parts of the body. You know that there are parts of our body which are more sensitive and which are able to larger variety, to greater variety of movements. Yes, these are hands, for example, and face, especially lips and tongue. Yes, and for example, genital organs. They have much more receptors than the other parts of the body. Yes, much more receptors, and that's why they occupy larger portion of this precentral gyrus. The same we can say, postcentral gyrus. The same we can say about motor function. Yes, motor homunculus. We can perform by our hands very precise movements and a great variety of movements, not like by our forearm, upper arm, not like by our upper limb. Yes, and so that's why our hands and also again tongue, lips, and face as a whole, it occupies a larger part of precentral gyrus. So the more receptors are located in a definite part of the body, the larger part of the cortex it occupies. And the more axonal terminals is present also, the larger part of the cortex of precentral gyrus, this part of the body occupies. So again, I'm telling you, such an arrangement is known as sensory and motor homunculi. Homunculus, it is singular. Homunculi, it is plural. The other centers about which we can talk, there is primary auditory area or projectional center of hearing that is located in the middle 1/3 of the superior temporal gyrus. There is primary visual area or projectional center of the vision, and it is located in the cortex on the medial surface of the occipital lobe around the calcarine sulcus. So it's here, projectional cortical centers of olfaction and taste, they are located in the cortex of uncus and perihippocampal gyrus. It's here. All of these centers, they are projectional centers. But I told you there are also associative, which are connected with corresponding projectional centers. For example, here in the posterior 1/3 of inferior frontal gyrus, there is speech area or Broca's area or motor speech area, we also can say. This Broca's area is responsible for this for ability of the person to speak. This associative center is located in posterior 1/3 of inferior frontal gyrus, not accidentally. It is so because primary somatomotor cortex, from where fibers to head and neck start, it is located in the inferior 1/3 of precentral gyrus. So due to the anatomic proximity, Broca's area is located here in the posterior 1/3 of inferior frontal gyrus. In case of any injury, hemorrhage, trauma of this area, person loses an ability to speak, but he doesn't lose an ability to understand the speech. He doesn't lose an ability to write, read. He can do all of the things, and he can pronounce sounds, but he cannot combine the sounds to form a word. Yes? In the posterior 1/3 of the middle frontal gyrus, there is graphia center or center of written speech. And also its location is not accidental because it's very close to the part of precentral gyrus from where fibers to upper limbs start. So in case of any injury of this region, person loses an ability to write but not speak. Yes? In the posterior 1/3 of the superior temporal gyrus, there is secondary auditory area and also Wernicke's area. It is area of comprehension of speech. Like such a person can speak himself, but when he hears somebody, another person, he cannot distinguish the words. Every time it seems to him that person speaks another language that he doesn't know. It is due to the damage of Wernicke's area. So there are many such associative centers, and you will discuss them, or you have already discussed them at your practical classes. I was just trying to explain you that cortex is functionally not homogeneous. Each part of cortex has a specific function that it tries to perform. So that was about the cortex. Gray matter is also presented by basal ganglia. So basal ganglia, I told you it is a misnomer. The correct name, it is basal nuclei. It is a group of subcortical nuclei which are situated at the base of the forebrain or telencephalon. Basal ganglia are strongly interconnected with the cerebral cortex, thalamus, and brainstem. And they perform the following functions. They regulate voluntary movement, and they provide cognitive function and emotional function. Totally, they are named basal because they are located on the base of the brain, yes? And so they are four. They are four. Here in the frontal section, we can see lentiform nucleus that includes putamen from lateral side and globus pallidus from medial side. Then laterally, sorry. Алло, да. Ну, у меня лекция. Угу. 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 Не знаю, впервые слышу от вас. Я же пропустила это все, надо у Светланы Ивановны спросить. Я же в конце только пришла. Угу. Ладно. Okay. So I told you, yes, lentiform nucleus that consists of putamen and globus pallidus, and then laterally from the putamen there is claustrum. Here we can see also caudate nucleus close to the lateral ventricles, and one more, it is amygdaloid complex that is located, that is surrounded by white matter of temporal lobe. In some books, you will find information that red nucleus, nucleus ruber, and substantia nigra, black substance, they also belong to basal nuclei. But we will not consider that. Actually, they are not located even in cerebral hemispheres. So they are functionally very similar. They perform the same functions. Yes, through red nucleus and through the substantia nigra, tracts which start from basal nuclei pass until they before they reach spinal cord and target organs, yes, working organs, but we will not refer them to basal ganglia. Just you have to understand that they are functionally interconnected. Here in the horizontal section, we again can see caudate nucleus, and we can see that head of caudate nucleus forms lateral wall of the anterior horn of the lateral ventricle, yes, and there is also lentiform nucleus that is located laterally from caudate nucleus and thalamus, and laterally from lentiform nucleus there must be claustrum, just it's not visible here. And amygdaloid complex in this horizontal section is not visible because it is located inferiorly, lower. So what are exact functions of each of the nuclei? Lentiform nucleus together with caudate nucleus they form corpus striatum or stria pallida system. I have already told you that lentiform nucleus consists of globus pallidus, that is medial part and it is more Asian part, and putamen, that is lateral part and it is phylogenetically newest part. So putamen, if to translate it from Latin, it will be like eggshell, like that thing which you should remove before eating egg. Yes. Globus pallidus, it is translated as a pale ball. Yes. So its color is lighter than color of putamen. We have talked about it already when we discussed cerebellum, when we were saying that cerebellum has 4 nuclei, and the most medial is the lightest, and the most lateral, that is dentate nucleus, it is the darkest, because phylogenetically it is the newest nucleus. It contains more neuron bodies. So here we can say the same. Globus pallidus is more Asian structure, and putamen is the newest. So it contains more neuron bodies. And caudate nucleus, together with putamen, they form striatum, and globus pallidus, it is pallidum. So together, striatum and pallidum forms corpus striatum or stria pallida system. So the stria pallida system regulates muscle tone, functioning of internal organs, and elaboration of conditional reflexes. Striatum, it is as newest structure. It has inhibitory action, and pallidum has stimulatory action. So in case of pallidum activity, movements become very active and very like the range of movements increases. And in case of striatum activation, movements are getting more precise. So in childhood, pallidum is more active. That's why small children are unable to perform precise movements. Yes, they cannot write, they don't have a good handwriting, they cannot collect puzzles, for example, because all of this, it requires very precise movements. And striatum gets activated with an age, when child grows up. When he is about 7 or 8, at that time he is usually able to perform precise movements. So function, yes, here again in this frontal section you can see globus pallidus and putamen here, and this is caudate nucleus, and together they form stria pallida system. Laterally from the putamen there is claustrum. Function of claustrum is still unknown. But what we can say that it looks like an age. If to translate it from Latin, it will be age, yes. And it consists of gray matter, of neuron bodies, but function is still unknown. Amygdaloid nuclei. Amygdaloid complex consists of numerous amygdaloid nuclei. Here you can see it in the temporal lobe, surrounded by white matter. So amygdaloid complex is a part of limbic system. And it is responsible for agonistic behavior and aggression and fear. Bilateral removal of amygdala in animals causes loss of aggression. So if to remove both amygdala from the brain, then animals will lose aggression, fear. From the one side, it's very good. Yes, they will become so cute, so friendly. But actually, aggression is a defensive is one of our defensive ability, aggression and fear. If we are not afraid of anything, and if we do not feel any aggression to anybody, then we cannot recognize a person who is tuned, not friendly to us. Yes, and so we lose an ability to estimate risk. So persons are getting risky, and they start taking risk of their lives, and usually such people do not live long. Yes, because if to take risk all the time, one day you will not be lucky and something will happen, yes, any accident. So actually amygdala is very important. Olfactory brain. So the oldest part of cerebral hemispheres, it is olfactory brain, and it has peripheral parts, peripheral and central part. So peripheral part is presented by olfactory bulb, and then posteriorly olfactory tract, and the most posterior part, the posterior part, it is olfactory trigone. Central part is presented by cingulate gyrus that continues to the parahippocampal gyrus, dentate gyrus, and uncus. So all of these four gyri, they form fornicate gyrus that surrounds as a fornix corpus callosum. Yes. So olfactory receptors or olfactory analyzer is the most ancient analyzer among all analyzers of special sensation. So it is the first feeling creatures acquired, yes, during evolution. That's why olfactory brain, all the structures of olfactory brain, they belong to limbic system. And limbic system is not only responsible for the olfaction, but it supports a variety of functions, including emotions, behavior, motivation, long-term memory, and olfaction. A collection of structures from the telencephalon, diencephalon, and mesencephalon comprises limbic system. So which exactly structures are included here? This is, yes, again, olfactory bulb. Yes, we have already seen it. Olfactory tract, olfactory trigone, anterior perforated substance, cingulate gyrus, parahippocampal gyrus, uncus, dentate gyrus, fornix also. Yes, and amygdaloid complex, hippocamp, medullary bodies, because if you remember, they are subcortical centers of olfaction, and so on. So there are very many parts of limbic system. And yes, they are responsible not only for the olfaction, but also for the emotions and some other things. Okay. So yes, what are exactly functions of limbic system? This is development and preservation instincts, eating, drinking, sexual activity, defense, formation of emotions and behavioral responses, organization of short-term and long-term memory, regulation of functions of internal organs, and participation in sleeping arrangements. Okay, that was about gray matter. White matter of cerebrum is presented by neuroglia, that is supportive tissue. Yes, it is present in each part of the brain, in each part of CNS, and also myelinated nerve fibers, which are axons. Yes, and so this myelinated nerve fibers, they can be divided into three groups. Associative fibers are those which connect cortex inside one cerebral hemisphere. Commissural fibers are those which connect two cerebral hemispheres with each other. And projectional fibers are those which connect cortex with lower parts of CNS. In the other parts of the brain and CNS, nearly all the fibers, they are projectional. We know, we have said about it, that there are conducting tracts, they move upward and then downward, so all of these are projectional fibers. But here, yes, there are some additional fibers. So association fibers connect cortical areas within the same cerebral hemisphere. Long and short association fibers are distinguished, so there are long and short. Short association fibers, or U fibers, connect adjacent gyri. And long association fibers connect more widely separated areas. Long association fibers. So examples of the short, yes, here you can see the short association fibers. We can see here, these are like arcuate fibers. They are present between each adjacent gyri, and long association fibers, they form different fascicles. So here we can see, for example, superior longitudinal fascicle that connects cortex of frontal lobe with cortex of occipital lobe. And there is uncinate fasciculus that connects cortex of frontal lobe with temporal lobe. Inferior longitudinal fasciculus that connects temporal lobe with occipital lobe. Then cingulum that connects frontal lobe with occipital and also temporal. So there are different types of long association fibers. Commissural fibers interconnect corresponding regions of two cerebral hemispheres. The largest structure that is formed by commissural fibers, it is corpus callosum. There is also fornical commissure that is between two crus of fornix. Fornix as itself is formed by projectional fibers because it connects mammillary bodies with hippocampus, parahippocampal gyrus. But there is this fornical commissure that is between two crus of fornix. It is formed by commissural fibers. There is anterior commissure also here below the rostrum of corpus callosum. There is posterior commissure, but it doesn't belong to cerebral hemispheres. It belongs to the diencephalon. Yes, posterior commissure or habenula commissure. Corpus callosum is formed by several parts. The most posterior part is splenium, then it continues to the body or trunk, then genu or knee, yes, and the last one, it is rostrum. So these commissural fibers, after they pass through this commissure, for example, corpus callosum, they continue moving laterally, and they form different structures, like between these fibers, which connect cortex of frontal lobes, it forms forceps minor. And those fibers which connect cortex of occipital lobes, they form forceps major. Commissural fibers interconnect both homologous and heterologous areas of the two hemispheres. So it's not compulsory that commissural fibers, they are between two frontal lobes. They can start from frontal lobe of one side and they can move to the parietal lobe of opposite side and so on. So this is the largest structure that is formed by commissural fibers. So projectional fibers connect cerebral cortex with lower parts of brain and spinal cord in both directions. And the largest structure that is formed by projectional fibers in cerebral hemispheres, that is internal capsule. Here we can see it, capsula interna. Internal capsule is located between caudate nucleus and thalamus medially and lentiform nucleus laterally. And it is divided into three parts. That part that is located between head of caudate nucleus and lentiform nucleus is named anterior limb or crus anterior. That part that is located between thalamus and lentiform nucleus, it is named posterior limb or crus posterior. And between them, there is a knee or genu. So it's V-shaped, anterior limb, posterior limb, and knee in between, yes? You have to know which tracts pass through each of these structures. So through the anterior limb, frontothalamic fibers pass, as well as frontopontine fibers. You remember that frontopontine fibers are those which start from the cortex of frontal lobe and then they move downward through the internal capsule, through cerebral peduncles of the midbrain, downward until they reach pons, where they form synapse with pontine nuclei, yes? And from the pontine nuclei, pontocerebellar fibers start, which enter through middle cerebellar peduncles, cerebellum. Through the posterior limb, corticospinal tract passes, and spinothalamic fibers, corticothalamic, Temporoparieto-occipitopontine fibers, the same like frontopontine, yes, function is the same, just they start from another lobes. And visual and auditory fibers, or optic and acoustic radiation. So those fibers which start from subcortical centers of vision and hearing, and they move to the cortical centers of vision and hearing. And through the knee or genu of the internal capsule, corticonuclear tract starts. We also can say that between putamen and claustrum, there is external capsule, capsula externa, and between claustrum and cortex of insula, there is extreme capsule, capsula extrema. And they contain also associative fibers. Conducting tracts, the other point which we have to discuss today. So conducting tracts are bundles of nerve fibers which connect functionally similar or different areas of gray matter in CNS. And they occupy definite place in white matter of brain and spinal cord. And they transmit the same impulse. So we actually have already discussed the things at our practical classes. We were saying that both gray matter and white matter, they are arranged. Yes, neuron bodies and processes, they are not chaotically disseminated. Yes, they form like neuron bodies, they form this nuclei. Yes, and processes, they form tracts. Yes. So, yes. So actually, it's not the first time when we are talking about it. All the conducting tracts, they are divided according to the direction of transmission into ascending or descending. So sensory or ascending and motor descending. Yes. According to the presence of fibers decusation, they can be decussated. If they are decussated, they can be decussated once or twice, and non-decussated. And according to the connection with cerebral cortex, they can be conscious if they are connected to cerebral cortex. So information reaches cortex and we realize what's going on. And when we move, it was our decision to move. Yes. And unconscious. These are reflexes. Yes. Or if tract is unconscious, then using it, unconditional reflexes is performed. So among the ascending tracts are classified according to type of the receptor. They can be exteroceptive when receptors are located in skin and visible mucosa. They can be proprioceptive when tract starts from proprioceptors, which are located in the organs of locomotor apparatus. And then can be visceroceptive tract when it starts from interoceptors. So the example of proprioceptive tract, it is conscious proprioceptive tract, it is a bulbothalamic tract that is found in spinal cord, that forms in the spinal cord fasciculus gracilis and fasciculus cuneatus. Fasciculus gracilis transmits sensory impulses from lower limbs, and fasciculus cuneatus transmits sensory impulses from upper limbs. It is located in the posterior funiculi and conducts sensory impulses associated with senses of touch, pressure, not touch actually, yes, deep touch only. So body movement from muscle, tendons, and joints to the brain. So for the bulbothalamic tract, when you talk about the tract, you have to know how many neurons it contains, where is each neuron body located, if it forms decussation or not, and that is all. Yes, and if there is a decussation, which order neuron forms this decussation and at which level. So for the bulbothalamic tract, body of the first order neuron is located in dorsal root ganglion. Actually, it is always like that for all the ascending tracts. Then axon of the first order neuron enters spinal cord through posterolateral sulcus, but it doesn't enter gray matter. It directly goes to the posterior funiculus, and it forms these two fascicles, gracilis and cuneatus, and they move upward until they reach medulla oblongata, where there are two nuclei, nucleus gracilis and nucleus cuneatus. These two nuclei, yes, here synapses are formed. Yes, axon of first order neuron forms synapse with body of second order neuron, and then axons of second order neuron decussate at this level, and they form medial lemniscus that passes through the pons, midbrain, reaches ventrolateral nuclei of thalamus, forms synapse with bodies of third order neurons which are located in the ventrolateral nuclei of thalamus, and axons of third order neurons, they reach cortex of postcentral gyrus. That is the main somatosensory area. So that is bulbothalamic tract. Usually, all the ascending tracts receive their names from the location of bodies of second and third order neurons, because body of the first order neuron is always located in the dorsal root ganglion. Okay. Unconscious proprioceptive tracts, they are spinocerebellar. So for spinocerebellar tract, yes, they are two, posterior and anterior. So again, body of the first order neuron is located in the dorsal root ganglion. Then body of the second order neuron is located in the posterior horn. Then for the posterior spinocerebellar tract, fibers do not decussate. They just enter lateral funiculus of the same site, moves upward until it reaches medulla oblongata, and then through inferior cerebellar peduncles, it enters cerebellum. For anterior spinocerebellar tract, axons of second order neuron decussate, and they move to the contralateral lateral funiculus, to the opposite side. Also moves upward, passes through medulla oblongata, pons, and midbrain, and then again decussates, and through superior cerebellar peduncles enters cerebellum. So posterior spinocerebellar tract is not decussated, and anterior spinocerebellar tract is decussated twice. Anyway, fibers end in the cortex of cerebellum of the same site. So that's why any injury in right cerebellar hemisphere, for example, will cause consequences again in the same site, in the right side. Okay, spinothalamic tracts, they are exteroceptive. There are two of them, lateral and anterior spinothalamic. Lateral spinothalamic tract is responsible for pain and temperature, and anterior spinothalamic tract carries impulses about touch and pressure. For them, actually, allocation of the neurons is the same. So body of the first order neuron is located in the dorsal root ganglion, then body of the second order neuron is located in the posterior horn, then fibers of the axons of second order neuron decussate. And for the lateral spinothalamic tract, they move in the lateral funiculus of the spinal cord, and for the anterior spinothalamic tract, they move in the anterior funiculus. Then in the medulla oblongata, they join, form spinal lemniscus, pass through the pons and midbrain, and again they reach ventrolateral nuclei of thalamus, which contain bodies of third order neurons and bodies and axons of third order neurons, and in the cortex of postcentral gyrus. So the only difference is in the location of axons of second order neurons in the spinal cord. For the lateral spinothalamic, they are located in the lateral funiculus, and for the anterior spinothalamic, they are located in the anterior funiculus. All the descending tracts are divided into pyramidal and extrapyramidal. So pyramidal tracts, I have already told you, they are conscious, they start from the cortex, and they are named pyramidal because they start from giant pyramidal cells in the precentral gyrus. So they are two, it is corticospinal tract and corticonuclear tract. All of them, yes, each of them. Okay, let's start with corticospinal. Corticospinal tract starts, its first order neuron is located in the cortex of precentral gyrus. and if to be more precise, in the fifth layer. And then it passes through the axons, pass through posterior limb of the internal capsule and move downward, passes through the base of cerebral peduncles and then pons and the medulla oblongata. In the medulla oblongata, it forms pyramid, yes? And at the level of pyramid decussation, 80% of fibers decussate, and they form lateral corticospinal tract that passes in the lateral funiculus of the spinal cord. An axon of first order neuron forms synapse with body of the second order neuron of the opposite side that is located in the anterior horn. Then axon of the second order neuron leaves through ventral root, through anterolateral sulcus, through the ventral root of the spinal nerve, and it reaches target organ. Usually it is skeletal muscle. Yes, not usually, it's always skeletal muscle. 20% of fibers do not decussate at the level of pyramid decussation. They continue downward in the anterior funiculus and they form anterior corticospinal tract. At the very end, at the level of the segment where axon of first order neuron forms synapse with body of second order neuron, most part of fibers decussates. But small amount of fibers does not decussate and it ends in the neuron bodies of the same site in the anterior horn. And then again, axons leave through anterolateral sulcus, through the ventral root of the spinal nerve until it reaches target organ. For the corticonuclear tract, it's all the same. Again, it starts from precentral gyrus, yes, and it passes through the knee of internal capsule, base of cerebral peduncles, and it forms synapses with motor nuclei of cranial nerves. So fibers of corticonuclear tract partially decussate, so that nearly all the motor nuclei of cranial nerves, they receive double supply from both sides. There are two exceptions, that is motor nucleus of facial nerve and motor nucleus of hypoglossal nerve, and it receives supply only from one side. There are also extrapyramidal tracts which do not start from the cortex of cerebral hemispheres. They are reticular spinal tract that starts from reticular formation of the brainstem, and it conducts motor impulses associated with maintenance of muscle tone and the activity of sweat glands from the brain. And then rubrospinal tract that conducts motor impulses associated with muscular coordination and the maintenance of posture from the brain. It starts from the red nucleus. And there are many of them. Yes, you have to read. But the previous one, which we have discussed before, you even have to be able to draw to pass the exam successfully. So meninges of the brain. Actually, you studied it when you studied meninges of spinal cord. Here they are the same. There are three meninges. It is dura mater and arachnoid mater and pia mater. And dura mater of the brain, unlike dura mater of spinal cord, it is firmly attached to the internal surface of skull bones. That's why, unlike in the spinal cord, here in the brain, there is no epidural space. Yes. So dura mater is the thickest membrane, meninges of the brain. Yes, it performs protective function, and also between two layers of the dura mater, there are venous sinuses. So cavities, like canals, through which venous blood outflows from the brain. This is additional function of dura mater that was not present in dura mater of spinal cord. Arachnoid mater is beneath, yes, under the dura mater, and pia mater is the innermost. Okay, so dura mater also forms processes which go into fissures between various parts of the brain, such as falx cerebri, into the longitudinal fissure between cerebral hemispheres, falx cerebelli between hemispheres of cerebellum, and tentorium cerebelli separates cerebellum from the occipital lobe. There are inner and outer layers of dura mater and spaces between them having a triangular shape in the section. They are called sinuses. So here in this diagram, for example, you see superior sagittal sinus. Arachnoid matter, it is the middle covering, and arachnoid matter enters the largest fissures. For example, it enters longitudinal fissure of the brain, but it doesn't enter small sulci. Pia matter, that is vascular tunic, it consists of numerous blood vessels, capillaries. It enters each sulcus, each even the smallest sulcus. Between arachnoid matter and dura mater there is subdural space, and between arachnoid matter and pia mater there is subarachnoid space. And because of difference in the course of arachnoid matter and dura mater, there are some enlargement of subarachnoid space, like extensions in some regions, and they are named cisterns. The peculiarity of the vascular membrane or pia mater, it is that in the ventricles of the brain, it forms vascular plexuses or plexus choroidos. What do you mean it's the same in spinal cord? What exactly is the same in spinal cord? Meninges are the same, but there are no sinuses, there are no cisterns. Yes? What do you mean? Okay. And now I want to show you the video about production and outflow of cerebrospinal fluid. Please take a look. Yes, you are right. Yes, meninges are the same, but they perform, like there are some additional functions which are not present in this meninges of spinal cord, and course is slightly different. But names are the same, yes. Actually, dura mater of the brain continues into dura mater of spinal cord. All of them continue into each other. That is right.
This is Dr. Cal Shipley with a review of cerebral spinal fluid production and circulation. Let's take a look at some anatomy first. The brain and spinal cord are completely surrounded by cerebrospinal fluid, also known as CSF. The CSF fluid layer acts as a mechanical shock absorber, cushioning movements of the brain in head trauma. The CSF also carries nutrients to and waste products away from the brain. Most of the cerebral spinal fluid is produced in a series of chambers known as the ventricles. The largest of these chambers are called the lateral ventricles. Rotating to a side view looking from the left, we see the lateral ventricle connected to the third and fourth ventricle. The third and fourth ventricles are connected by a narrow passageway known as the aqueduct. Obstruction to flow of CSF within the aqueduct is one of the most common causes of hydrocephalus. The brainstem represents the lowermost portion of the brain and connects the brain to the spinal cord. Here we see a close-up view of the relationship between the aqueduct, the fourth ventricle, and the brainstem. As can be seen here, the aqueduct actually lies within the substance of the brainstem. The majority of cerebral spinal fluid is produced within the lateral and third and fourth ventricles as a result of specialized tissue known as choroid plexus. The functioning of the choroid plexus is yet another beautiful example of the adaptive physiology of the human body. Here we see the typical position of the choroid plexus in the lateral and third and fourth ventricles. The cerebral spinal fluid is not produced by the cells of the choroid plexus. Instead, CSF represents a filtrate of the blood, with the filtration performed by the highly specialized cells of the choroid plexus. The functional unit of the choroid plexus is known as a villus, or multiple villi. The villus consists of loops of tiny capillaries separated from the ventricle by a layer of cuboidal epithelial cells. Here in a close-up view we see the layer of cuboidal epithelial cells interposed between the capillary and the ventricle. Like all capillaries, those of the choroid plexus have a wall which is a single cell thick. This allows for ready transport of ions and molecules to and from the capillary. The cells of the cuboidal epithelium are highly specialized and are capable of generating a polarized electrical charge across their membranes. This charge causes sodium, chloride, and bicarbonate ions to migrate from the capillary plasma into the ventricle. This ionic flow creates an osmotic gradient, which draws water molecules from the capillary plasma into the ventricle. And so the composition of cerebrospinal fluid is mostly water with sodium, chloride, and bicarbonate ions, and a smattering of other ions, as well as small amounts of amino acids, protein, and glucose thrown into the mix. The filtrate contains no cells, but under normal circumstances, small numbers of white blood cells, usually monocytes, are introduced into the cerebrospinal fluid directly from the vascular system, once the CSF is within the ventricles. And so the cerebrospinal fluid originates in the ventricles as a result of filtration of the plasma by the choroid plexus. It then flows or circulates through the ventricles, then surrounds the brain and spinal cord, where it is contained by a layer of the meninges known as the arachnoid. Through a series of outpouchings on the arachnoid known as granulations, the cerebrospinal fluid then flows into the venous sinuses of the brain, returning to the bloodstream from whence it originated. The total volume of cerebrospinal fluid at any given moment is about 150 cc's in an adult. Approximately 600 cc's of cerebrospinal fluid is produced each day via blood filtration in the choroid plexus. So if you do the math, 100% of the volume of cerebrospinal fluid turns over every 6 hours, or 4 times a day.
Okay, so that was the video. I think it explained you very clearly how cerebrospinal fluid is produced and where it outflows. That is all I wanted to tell you for today. Do you have any questions? There are many parts of the brain which are responsible for sleeping. We were talking about it at the previous lecture, like we can say pineal gland participates, yes, because it produces melatonin, and reticular formation, especially locus coeruleus, it is also responsible, and limbic system somehow also participates because it also participates in sleeping arrangements. Like, it is a complex process. We cannot say that only one part of the brain is responsible for it. Like, all of them interact with each other. Any other questions? Okay. Thank you. Same to you. Okay. If you have no more questions, then you may be free. See you next time. Goodbye.